This is the first cell of a future multicellular organism. It begins to divide, resulting in a cluster of identical cells that amazingly transform into a complex organism. Today we are going to talk about how this happens. I'm not a biologist, so I might have some inaccuracies in describing the process. My primary interest lies not in specific details but rather in the underlying mechanisms and algorithms governing the formation of multicellular organisms. How complexity arises from simple rules. That's what this project is all about. In our previous video, we created a simple shapeless multicellular organism starting with one cell. There, I described in detail how cells interact with each other and determine their developmental path within the organism. Today, we are going to create an organism with structure and symmetry typical of bilaterally symmetrical animals. We'll focus only on the general body plan. The organism will consist of just 3 types of cells: bone, flesh, and skin. Manually creating something more complex would be difficult. Only evolution can achieve such unimaginable complexity. We'll discuss this in the second part of the video. For proper structural development, the embryo must be divided into segments along its body axis. Sometimes these segments are clearly visible. In vertebrates, they're usually called somites, and segmentation is evident in spinal column structure. Let's consider how segmentation occurs using the example of the fruit fly Drosophila. Embryonic segmentation proceeds through morphogen gradients. However, since morphogens cannot spread over long distances, and a small embryo can't be divided into many segments, so segmentation takes place incrementally as the embryo grows. At each stage of segmentation, different sets of genes are involved. Then, the segments are distributed among several groups, and in each group, a specific Hox gene becomes active. I'll explain more about them in this video. Cells in the embryo can move, vary in size, and divide at different rates. This allows for arbitrary bending of cell layers to achieve the desired result. All this takes place in 3-dimensional space, forming various structures during growth. Most of these capabilities are unavailable in my model. Let me outline its limitations. My model is 2-dimensional. All cells have fixed sizes and positions on a hexagonal grid, meaning no bends or curves are possible. Cell division is restricted to those located at the edges. Therefore, if we segment the embryo initially, proportions won't remain consistent during further growth. So, segmentation in this model follows a slightly different approach. There are organizer cells, which play a key role in forming the organism's structure. For instance, they secrete morphogens that define boundaries for other cells' growth. Or morphogens to mark segment borders. Besides influencing neighboring cells via morphogens, organizer cells can also affect individual adjacent cells by activating or deactivating certain genes. By doing so, organizer cells establish guides that ultimately determine the shape of the developing organism. Here, an organizer cell crosses beyond the blue morphogen boundary, marking the start of a new segment. Now it also produces a morphogen specifying the border of the new segment. This method will be used for initial segmentation of the embryo. Organizer cells can create guiding paths not only straight but also split into 2 or 3 branches. With the help of a particular morphogen, unnecessary branches can be prevented from forming. It's time to design a genome that allows us to develop a complex, bilaterally symmetric organism from a single dividing cell. This is the first cell. I've programmed the initial genetic code, and will begin the process. The first cell initiates a guideline. All guideline cells produce a morphogen marked yellow here. Within this morphogen field, other cells can divide. The red-marked morphogen is used for initial segmentation. You can see a similar process in the top left corner of the video, where somites form along the neural tube of a vertebrate animal embryo. Now we have an elongated embryo divided into multiple segments. Each segment is divided into two halves. These will serve as the basis for vertebrae and intervertebral spaces. As the embryo grew, I gradually reduced the activity of a specific gene in the cells until it reached zero, stopping further growth. Additionally, while a segment was growing, I gradually decreased the activity of another gene. When forming a new segment, if the activity of this gene had dropped to zero, I reactivated it fully. This allowed larger sections to emerge in the embryo. Within each section, a unique Hox gene became activated. A Hox gene produces a transcription factor, a type of protein responsible for regulating the expression of other genes. Any cell can identify its location in the body based on the activity of Hox genes. In this project, like in flies, there is only one set of Hox genes. During evolution, complete duplication occurred a couple of times, resulting in 4 sets of Hox genes in mammals today, whose functions diverged over time. Having more Hox genes allows for far greater complexity and detail in body structure. Currently, all zones have equal length, but by referencing Hox genes, we can adjust the rate at which we reduce the activity of the gene used to divide the embryo into zones. Now each zone has a different size. Guideline cells also release a morphogen marked red here. Upon differentiation, under the influence of this morphogen, cells form bones. Our current structure looks somewhat rectangular. To smooth out the tail and head, let's use the same gene we've been using to determine zone dimensions. Proportionately to the activity level of this gene, we'll release a morphogen serving as a boundary for lateral extensions of the guidelines. This results in a gradual tapering of the tail thickness. Almost the same trick applies to shaping the head, except here both the segment size and thickness decrease smoothly. Next, cells in appropriate locations will emit a morphogen inhibiting bone formation. Furthermore, we'll shrink 2 segments so that the bones fuse together, forming the foundation for shoulder girdle and pelvis. The head and tail are ready. It's time to address the central part. Since each zone has its own active Hox gene, setting conditions is straightforward. Limit lateral guideline growth in the neck area. Increase growth in shoulder and hip areas. Enhance growth in torso while implementing a gradual reduction in size. Block bone formation near lateral guidelines in lumbar region. One final touch remains: adding limbs. Similarly, here everything divides into zones, each controlled by its own active Hox gene. It's important to understand that a Hox gene doesn't carry information about how a particular body part should look like. That's handled by entirely different mechanisms. Offspring of this organism will have limbs marked by the same Hox genes, homologous down to having the same set of bones, yet potentially very different shapes. By designing a single limb, I obtained all limbs for the organism. Previously, this wasn't obvious, and it was believed that somewhere there were blueprints stored separately for each limb. Over the course of evolution, they may change to better adapt to the environment. But they'll still be mapped by the same Hox genes. Both forelimbs and hindlimbs utilize the same Hox genes, but the rest of the regulatory genes could differ significantly, leading to divergent evolutionary pathways. I've designed the limb. Time to initiate embryonic development. What emerged isn't quite what we expected. The issue is that limbs are symmetrical, and for correct formation, the limb bud needs to know its anterior-posterior orientation. In tetrapods, a gene at the posterior margin of the limb bud is activated, producing a signaling morphogen whose gradient defines the axis. On the right side, you can see a chicken embryo's wing when the same gene was activated at the front margin as well. Issues with this gene's expression can lead to abnormal development. In my project, it was easier to create a gradient from the front edge of the limb. I've highlighted this morphogen in pink. It activates in cells a gene producing the very same morphogen. This allows it to propagate throughout the organism. Activation occurs only at the edge cells unless blocked by another morphogen, shown here in yellow. This prevents the pink morphogen from crossing over to the opposite side of the limb. Ultimately, we ended up with a fine specimen. Now I'll tell you about one mechanism that likely contributes to laying down axes for future fingers. We utilized a gradient to define the direction of limb curvature, whereas all fingers are identical and their number is rigidly predetermined. How nature forms finger axes remains less clear. In my model, morphogens don't react to each other, but in reality, they actively interact. In 1952, Alan Turing proposed a reaction-diffusion model describing interactions between two substances' morphogens. This model generates complex patterns depending on the parameters of these substances. This model effectively explains the diversity of patterns found in nature, and it's hypothesized that the same mechanism participates in establishing axes for future fingers. Experiments show that disabling Hox genes in mouse embryos leads to shortened fingers, fewer phalanges, and the formation of a cartilage arch connecting fingertips. Decreasing the activity of genes determining limb polarity increases the number of digits. Completely switching off both types of genes makes the limb resemble shark fins or primitive ray-finned fish appendages. All these phenomena fit well with models based on reaction-diffusion mechanisms. In the previous video, I delved deeply into regeneration processes. Let's try transferring it to this organism. However, here it causes some issues. If the required cell type is missing, the wound will fill with cells of another type that happen to be nearby. This problem could be solved by leaving stem cells capable of differentiating into the required cell type. In humans, this kind of process occurs where constant cell renewal is needed, such as in the blood system, intestinal epithelium, or skin. It's worth mentioning an important fundamental discovery in embryology. The discovery of embryonic induction became one of the key milestones in developmental biology, earning the Nobel Prize in 1935. It demonstrates that special cells act as organizers, directing the development of surrounding cells. For example, in one experiment, cells from a specific region of a newt embryo were transplanted into another embryo, causing the formation of a secondary axial organ complex on the same embryo. We replicated these experiments in our model by copying an organizer cell to a new location within the embryo. Limb development can be initiated on the head, though realistically, problems with brain connectivity would arise. Something similar can be observed in mutant flies, where legs grow on their heads. However, this involves a different mechanism. In these cases, pre-existing protrusions like antennae exist, and dysregulation leads to alternative development pathways. Recall that every cell contains the same genome, and cells in antennal locations inherently know how to form limbs properly. And now we're going to play with mutations. I tried introducing random mutations to observe what changes occur. Of the 32 organisms, 5 are clearly non-viable. Every sixth organism has no changes at all. That surprised me the most. Some organisms have minor changes. A thinner tail, a massive neck, thin fingertips, variations in limb lengths. 2 more mutations are similar to what we see in cetaceans and snakes. Who didn't know, whales and snakes belonged to tetrapods and their ancestors had limbs. It's difficult to say anything about a few more mutations, but considering what evolution did with skeletal systems in various vertebrates, these variants might prove viable. Pay attention to the cartilaginous fins of the rays. And the most interesting option is an organism with tentacles instead of limbs. Now let's deliberately introduce changes into the regulatory regions of the genome. Let's eliminate the tail, demonstrating that this can be achieved through mutations in different genomic locations. There's a gene in the tail itself whose activity diminishes gradually, leading to its narrowing. Another gene, whose activity decreases across the full body length, exists to restrict overall organism length. Thus, limiting tail length can be done by manipulating either gene's decreasing activity. First, we'll modify the gene controlling total organism length. Here's a great outcome. We've successfully eliminated the tail. Now let's attempt modifying the second gene. We'll do this in 3 versions to obtain varying tail lengths. Surprisingly, something unexpected happened. Let's figure out why. Hox genes possess an interesting property. They're often arranged in DNA in the same order they activate during organism growth. I utilized this feature, automatically enabling the next Hox gene once the preceding one's domain ends. Thus, upon activation, the gene responsible for limb development turned on unexpectedly in the new context. Nonetheless, I'll fulfill my task and demonstrate a few mutations ensuring our organism remains tail-free. We'll exploit the gene controlling overall body length. What happens if, under certain conditions, a Hox gene switches not to the next one but to any arbitrary gene? In this case, we're dealing with a gene whose activity declines across the whole organism's length. Until the activity of this gene drops below a specified threshold, the Hox gene won't switch to the subsequent one. Making slight modifications yield centipedes of any length. Minor alterations in regulatory regions operating during embryonic development lead to profound phenotypic changes. This aligns perfectly with one concept in evolutionary biology. Due to minor changes in gene regulation during embryonic development, mutants with dramatically distinct phenotypes can appear, despite being genetically identical and able to crossbreed freely. No simultaneous mutation occurrence in both male and female individuals is thus required. Point mutations ensure offspring inherits one phenotype or another. If vacant ecological niches exist, for example, after mass extinction events, this opens opportunities for rapid speciation to fill these vacant niches. Over time, 6-legged creatures might spend more time in trees, wide-tailed ones migrate to water habitats. Different phenotypes will encounter each other less frequently, evolving independently, eventually leading to reproductive isolation. Fossil records will reflect sharp transitions in species diversity without intermediate forms. This described scenario embodies the theory of punctuated equilibrium. Unlike classical views suggesting gradual evolution, this theory posits that evolution progresses through brief periods of swift change interspersed with prolonged stability phases. Most likely, both scenarios coexist. Our 6-legged creature faces prolonged adaptation for arboreal living, yet fossil records won't capture this process. After releasing the first video, viewers commented that the algorithm seemed overly complicated and convoluted. In fact, it's simple and logical because it was designed by human intelligence. In my first artificial life simulation, I employed a circular sequence of numbers as the genome. When reading data from the genome, the first number encoded a command, followed by arguments and jump addresses. Depending on the execution result, different jump addresses were used. Ultimately, we could proceed to the byte previously functioning as an argument, but now encoding a command, while prior jump addresses transformed into arguments. In the next cycle, the number acting as a jump address or argument earlier might become a command. Writing genome code in such style would be nearly impossible, exceeding human cognitive capacity. Yet, in simulations, artificial life evolved beautifully, generating intricate forms. While preparing the video, I discovered that a similar phenomenon actually exists in biology and is called ribosomal frame shifting. That's why I love biology. Even if you come up with something creative, it turns out that nature has already implemented it. 3 letters typically encode one amino acid, but shifting the reading frame by 1 or 2 letters allows extracting multiple proteins from a single RNA strand. Viruses exploit this technique to pack more genetic information into smaller volumes of genetic material. Similar occurrences also take place in mitochondrial DNA in humans. There, 2 overlapping genes share the same region, yielding different proteins. It's hard to believe that such complexities resulted from intelligent design. Random emergence appears much more plausible. The thing is, evolution operates essentially as an optimized trial and error process. We call something complex when it's difficult for us to understand. But the evolution algorithm doesn't try to understand the genetic code. It simply changes letters and sees what happens. The concept of complexity is unknown to it. Because it relies heavily on iteration, this method consumes significant resources. Programmers rarely employ genetic algorithms in practice since, in most situations, a more suitable algorithm can be devised. However, the beauty of evolution is that it requires no creator. It emerges spontaneously under appropriate conditions as a natural process. If there are replicators that produce imperfect copies of themselves and resources are limited, then the process of evolution is inevitable. Looking at the complexity and tangled nature of the code helps distinguish whether it was designed intelligently or arose through trial and error. The genetic code I created in the previous video looks something like this. Now I'm going to outline some thoughts regarding the emergence of complexity. Imagine bacteria need to begin synthesizing a specific protein after receiving a signal, delayed by a specified interval. The simplest solution, a signal activates the first gene, whose product slowly accumulates inside the cell. Once reaching a certain concentration, it triggers the target gene. That's its simplest and most obvious resolution. Suppose we have a population of bacteria employing this mechanism. Meanwhile, another bacterial population exists nearby, where everything works differently. Both populations perform the same function, but the second option seems unnecessarily complicated. Now imagine environmental conditions change. Suddenly, the second variation seems advantageous. Mutations occur randomly, and the second version simply offers more potential points for regulation, increasing the likelihood that this population adapts faster to changing circumstances. Over time, natural selection favors networks capable of finely tuning protein production. Thus, more complex networks can adapt quicker to changing environments, suggesting that increased complexity can indeed be favored by natural selection. This is currently a thought experiment, but it's easy enough to construct a model to test it, which I intend to pursue sometime soon. I'd like to showcase the variety of tools available to natural selection. In this project, I'm utilizing only one of many possible methods of regulation. I'll briefly describe a couple more options. Once information is read from DNA, we obtain RNA. However, before protein synthesis, several segments called introns must be removed from the RNA molecule. Remaining RNA fragments are spliced together, allowing protein synthesis. Certain conditions may disrupt this process, causing RNA segments to join incorrectly. This enables producing numerous diverse proteins from a single gene. Such a mechanism is termed alternative splicing. Returning to our example. Presence of a specific morphogen could trigger a different protein output from a gene, thereby creating an additional pathway to reach the target gene. There's also a brutal way to regulate the genome. Even if the entire chain of reactions fires correctly, the final RNA transcript might bind to another active molecule, preventing protein synthesis altogether. Thus, natural selection possesses a wealth of tools enabling precise regulation of gene expression. Genetic codes aren't rewritten intentionally, New fixes accumulate atop old ones, complexity builds naturally up to certain limits where drawbacks start outweighing benefits. Yet even this achieved limit astounds our imagination. That's all for today. Hit that like button, subscribe, leave a comment. Thank you to everyone supporting me on Patreon. Goodbye everyone.